So fatty acid oxidation is going to be happening, of course, in the fasting state. Because the fatty acids that are going to be taken up by cells for the beta oxidation process are going to be released from adipocytes due to glucagon signaling, and the fatty acids that are released from adipocytes will be using albumin as their carrier. So let's pick up at the fatty acid albumin carrier in the blood. This, of course, will be internalized into cells, this is the plasma membrane, by the same transporter that we've seen before. This is the fatty acid transport protein, FATP. Once the fatty acid enters the cell, we are now going to couple this with coenzyme A using the fatty acyl-CoA synthetase enzyme. And this, of course, will trap the enzyme and prevent any reverse transport out of the FATP transporter. Now this FA fatty acid CoA structure, of course, will be bound up by a fatty acid binding protein in order to shield the hydrophobicity from the cell and protect the cell, and also escort the FA CoA, fatty acid CoA, to the place where it's going to perform the beta oxidation reactions. And this, of course, is the mitochondria. So beta oxidation is going to occur within the matrix of the mitochondria. So let's draw primitive structure of a mitochondria and take a look at how the fatty acid is going to enter the mitochondria. If this is our outer mitochondrial membrane, this is our inner membrane, this is the inner membrane space. Recall that the outer membrane allows transport of larger molecules coming through, but what we also have associated with this outer membrane is an enzyme called CPT1. CPT1 stands for carnitine palmitoyl transferase type 1. The job of this enzyme is to convert the fatty acyl-CoA into a fatty acyl carnitine. Carnitine is a small molecule that we can get either from dietary source or we can synthesize it from the amino acid lysine. Once we form the carnitine structure, fatty acyl carnitine, of course we'll lose our CoA here. Now we have the molecule prepared for transport into the mitochondria. So on the inner mitochondrial membrane, passing the inner, we have a transporter. This carnitine now can transport, using this transporter, into the matrix of the mitochondria. This transporter is called the carnitine acylcarnitine translocase. And this specifically takes the fatty acyl carnitine structure, transports it in the mitochondria. Now once we've reached the inner mitochondrial matrix, we have another enzyme called CPT2. The job of this enzyme is to take the fatty acyl carnitine that has entered the mitochondria through the translocase and convert this back to fatty acyl-CoA. The reason for this is because the fatty acyl carnitine is not a substrate for fatty acid beta oxidation. fatty acyl-CoA is. So in this reaction, we will release carnitine. Now, in order to prevent a carnitine deficiency in the cytoplasm, we have to export the carnitine back out to continue rounds of fatty acid uptake into the matrix of the mitochondria. So the translocase transporter is a 1-to-1 antiporter. This means that the translocase will bind one molecule of fatty acyl carnitine in the intermembrane space, one molecule of carnitine in the matrix, and flip, allowing a 1-to-1 transport to restore carnitine into the cytoplasm where we can continue fatty acid uptake. Now, why is this process so important? Because fatty acyl-CoA cannot build up in the cytoplasm. If it does, especially in skeletal muscle, we'll have fatty muscle, and this will cause problems with insulin sensitization. So this process of transferring the CoA group with a carnitine group and then taking up the carnitine and re-establishing fatty acyl-CoA is a reaction that will essentially thermodynamically drive the fatty acyl group, fatty acid, into the matrix of the mitochondria by this reaction. This is all based on that substrate-product relationship. The product of one reaction disappears because it's the substrate of the following reaction, and so that will thermodynamically drive the free energy process into the matrix of the mitochondria. Now, once we have the fatty acid, fatty acyl-CoA structure in the mitochondria, let's take a look at the beta oxidation process, and let's take a look at this in a very rudimentary, easy way. Rather than look at the actual reactions that occur, let's see what happens here. So recall our nomenclature of a fatty acid. This is our alpha carbon. This is our beta carbon. So beta oxidation means that we are going to perform oxidative reactions on the beta carbon. The reason we need to do this is because we are going to release 2 carbon units, and this 2 carbon units will be released in the form of acetyl-CoA. So the reactions we're going to be performing on this for beta oxidation are essentially the reverse reactions that we use to synthesize fatty acids in the liver. Now, this reaction here, the oxidative reactions on the beta carbon, will be necessary in order to turn the beta carbon into a carbonyl. So this carbon was the beta carbon before we performed our series of 4 reactions to release the acetyl-CoA, and now this becomes the new alpha and new beta carbon, and now we perform oxidative reactions again on here to release the next 2 carbons as acetyl-CoA. Because we're in the matrix of the mitochondria, these acetyl-CoAs that are generated can enter the TCA cycle by condensation reaction with oxaloacetate to make citrate. Now let's take a look at the density of energy. The most common fatty acid that we have is a C16 palmitate. This molecule will go through 7 rounds of the 4 reactions necessary to release acetyl-CoA and will generate 8 acetyl-CoA molecules. Compare this to glucose. which will generate, in the end, 2 acetyl-CoAs. So 1 molecule of glucose generates 2 acetyl-CoAs. 1 molecule of C16 fatty acid will generate 4 times as much energy. It is for this very reason, the difference in energy capacity of these molecules is why dieting is so difficult. If we just had to exercise enough to burn the energy of 2 acetyl-CoAs, that would be a lot easier than the increase 4 times as much exercise in order to burn an equivalent molar amount of fatty acid. Now these processes here to do beta oxidation are the same. The reactions are the same in skeletal muscle and liver. With skeletal muscle, the job of this reaction is to simply generate ATP, energy, because this energy is necessary for contraction, and that's pretty much the major job of skeletal muscle. Now the liver, we have to consider one more event, and that's to prevent a futile loop. So let's shift over to the fed state in liver. Assuming that our glycogen stores are full, our energy charge in the liver is as high as it can go, then the rest of the glucose will be processed into fatty acid synthesis. Recall the last step of the fatty acid synthase complex to release the fatty acids from the complex is to do this by a thiolase enzyme to attach a coenzyme A. Now the question becomes, since there's a lot of mitochondria present in the hepatocyte, what prevents the fatty acyl-CoA from entering the mitochondria for beta oxidation? This would be a futile loop in the fed state in the liver. We make the fatty acid from glucose, it enters the mitochondria, we break this down by beta oxidation, and we lose all the energy that we've stored for the fatty acid. Well, recall that acetyl-CoA in the cytoplasm, which is generated from citrate lyase activity, generating oxaloacetate and acetyl-CoA. This is the first step of getting the components for fatty acid synthesis out of the mitochondria into the cytoplasm. The committed step into fatty acid synthesis, which is also the rate-limiting step, is to convert the acetyl-CoA into malonyl-CoA by acetyl-CoA carboxylase. Now recall this enzyme is activated by insulin signaling because we are in the fed state, and so we are driving the synthesis of acetyl-CoA into malonyl. Now when we take our malonyl-CoA together with our first acetyl-CoA, we are eventually going to make a fatty acyl-CoA molecule. In the liver, we have another role for the malonyl-CoA, and this is to inhibit CPT1. So elevated concentrations of malonyl-CoA, which are required for fatty acid synthesis, this is a substrate for making the fatty acid, but it's also an inhibitor of CPT1. So in the fed state with elevated concentrations of malonyl-CoA, we block CPT1 activity and prevent the newly synthesized fatty acyl-CoA from entering the mitochondria through this carnitine shuttle system. The fatty acyl-CoA that we make, of course, will be used for VLDL synthesis, which will be secreted from the hepatocyte. In the fasting state in the liver, acetyl-CoA carboxylase is inactivated because of glucagon signaling, so the conversion of acetyl-CoA to malonyl-CoA stops. Reducing the concentration of malonyl-CoA, we alleviate the block on CPT1, and so therefore in the fasting state in the liver, we can now enter fatty acyl groups into the mitochondria for beta oxidation.